What are some of the challenges that you see when it comes to kind of gathering this data and maintaining it for the future? I'm asking this because we talk a lot about machine learning being used to analyze data from past
Mhm.
clinical trials and the issue with standardization of data and things of that nature. So what are some of the gaps you see in the process or
Mhm.
areas that you hope to
Yeah.
see improved?
Yeah. So one of the things that comes up for us is we haven't really gotten as much into the machine learning aspect because it's so rare. We have so few numbers to really work with. But those same gaps exist in this area too without the machine learning in terms of, okay, is everybody using the same instrument? We need to kind of have a set standardized way that we do things, and then are they all trained to do it the exact same way? So that's something that we're working on too, is trying to streamline and standardize our approaches too. So that is an aspect that is ongoing, but something that we're working on. Okay, so how do you make sure that the data at this center is going to be the same as the data at that center is one big piece of it. And then some of the other things that come up too is, well, do we have um, enough kinds of tools to measure what we want to measure? So we have some rating scales that we'll use, but what else do we need? Do we have other kinds of ways to measure outcomes? Are we really getting at the problems that matter to families and the patients? And so that's an area that we're also looking into. We've put in for a grant. We'll see what happens with it. But one aspect of it was, okay, well, how do we quantify things like gait? Because being able to walk can be a really important thing for somebody's function. And so, can we better quantify gait changes and things like communication? So, language changes, that's really important to be able to speak to your child. And so, how does communication change over time? Can we quantify that too? So, that's a gap in terms of how do we measure things and how do we make sure that it's standard across centers and that's as objective as possible too. Another big issue is biomarkers. There aren't really great biomarkers in our area of diseases in the NCLs, and that is a hindrance to trial design, to figure out how do we see if something's responsive without having to watch them over 4 or 5 years before you decide. How can you get a quicker answer, so to speak, with a biomarker?
Has the incidence and prevalence of these diseases changed at all over the years? Has there been anything kind of more correlated with them now more than ever?
I'm not sure if it's truly changed or if we're just better at identifying them now. I think that it's probably more that genetic testing, it used to be so hard to get. You would do like a single gene test, you would have a high suspicion for it. And now it's pretty standard to say, we have this child who comes in with seizures and we don't know why. We're going to test. We're going to do a whole bunch of genetic testing on this child with a pretty broad panel. So I think we identify it more than we were before. And then the other piece that we still need to understand more and explore more is different phenotypes. So you have kids that come in, they might have the classic symptoms and then you test them to figure out if they have the disease. But what about people who don't follow that exact path? They either have a later onset or it's a little slower. I think that there's more to learn from that, because I know I've seen a couple of adults recently who have these diseases that we beforehand had typically thought to start in childhood, who are presenting with adult onset. So I wonder too, how many adults out there have had these odd symptoms that just could never really get, like, nobody could really figure out what they were, and then
Symptoms
they
like
just ended up, at some point progressing from a disease of unknown, of uncertainty, of no diagnosis. So I think there's some of that too.